Experimental controls are independent units and are not affected by experimental design. They are specific to the type of study or experiment. Researchers use controls to isolate variables of interest in a study, to identify background noise, to maximize detection of subtle treatment effects, to act as a standard against which unknown samples can be compared. Controls for Western blot include positive controls, negative controls, and internal loading controls. Positive controls is a sample known to endogenously express your target protein at physiological levels. They are used for optimizing reagents, validating antibodies, and are used as a control for recombinant proteins. Sources of positive controls include cell lines and tissues, transfected cells with epitope-tagged target proteins such as GFP, MYC, or His tags. Inadequate positive controls include overexpressed protein lysates, purified proteins, excess of target protein in a sample or purified proteins does not mimic a complex biological sample due to the imbalance in the ratio of target protein versus other proteins in the sample. The antibody selectivity cannot be properly evaluated. Negative control is a sample that does not express your target protein. They are used to test nonspecific binding of antibodies, to identify false positive reactions. Sources of negative controls include knockout animal tissues, cell lines generated using CRISPR, siRNA, drug-treated cell lines to inhibit protein expression. Inadequate negative controls are empty lanes, loading buffer alone, no primary antibody controls. No primary antibody controls for the specificity of the primary antibody, but do not control for the target protein in question. Internal loading controls ensures equal loading of experimental samples in each well of a protein gel. Ensuring equal loading is important for accurate comparative analysis of the changes in target protein expression. There are three different types of internal loading controls: housekeeping proteins, total protein staining, and pan proteins for phosphorylated proteins. Internal loading controls help in ensuring equal loading of protein concentration in each well and can be used to quantitate target proteins. Housekeeping proteins are proteins that are endogenously expressed and are unchanged by experimental design. Examples include GAPDH, COX-4, beta-actin. HKPs need to be confirmed to be unchanged by experimental conditions and should be in the same linear range as a target protein. Total protein staining detects total protein present in the sample. Examples include REVERT, total protein stain, Cipro-Ruby stain, stain-free technology. Measurement of the total protein signal and phosphorylation status of the target is important. Normalization of the phosphoprotein to its pan proteins allow the ratio of the phosphorylated protein to be evaluated accurately. And using the pan protein serves as an internal loading controls for higher accuracy. This is the best practice only in the case where pan protein expression is unaffected by the experimental treatment. Examples include pan-AKT, phospho-AKT combinations, pan-ERK1, phospho-ERK1 combination.